Okay, so hello, I'm Blandine Billet, and today I will present my work, my PhD work about the use of post-transition and transition metal ions to develop an APTA sensor. So thanks for the opportunity. This work has been done under the supervision of le Département de Pharmacochimie Moléculaire, the DPM, and the University Hospital of Grenoble in France. It is known for many years that metal ions can interact with nucleic acids in various ways. Their primary mode involves non-specific fast electrostatic interactions with the negatively charged DNA backbone. Then more specific interactions can occur with phosphates and nucleobases, such as outer sphere metal binding through a water molecule, inner sphere metal binding directly between the metal ion and the phosphate oxygen, or macrochelates when the bond is between the metal ion and the nucleobase. These interactions affect the structure and the conformation of nucleic acids according to several parameters. The nature of metal ions, like the charge, the size, the ligand exchange rate, for example, the nature and the structure of nucleic acids with some specific motif like G quadruplexes, thymine, or cytosine. Also, the experimental conditions like ionic strength or pH may play a fundamental role. To have a better idea of how metal ions interact with nucleic acids, here is a classification according to their preference for base over phosphate binding. To summarize, there are soft metal ions like silver or mercury that prefer end-donor atoms of nucleobases, leading to the destabilization of DNA double helix. Whereas hard metal ions like magnesium prefer O-donor atoms like phosphate oxygens, resulting to the DNA duplex stabilization. In between, there are borderline metal ions that have ambivalent properties on nucleobases and phosphates. In Aptamers field, alkali and alkaline RS metal ions are widely used, especially in buffers, due to their essential role in structural stabilization and in the formation of aptamer target complex. On the other hand, transition and post-transition metal ions are less employed. Some of them could be used as an alternative to the usual alkaline horse ions to stabilize some aptamer structures, but they are mainly considered as interferences in the development of fluorescence-based APTA sensor. Nevertheless, it is this group of elements that will attract our curiosity. So we choose to exploit the properties of transition and post-transition metal ions to induce significant disrupting effects on nucleic acid structure in order to develop an APTA sensor based on the DNA switching activity from an inactive state where metal ions are complexed to the aptamer to an active state where aptamer is bound to its target. This concept involves electrostatic interactions between the anionic fluorophore and the metal cations bound to the aptamer. So we used fluorescence anisotropy as a readout mode since it's a technique widely used in small molecule analysis. The advantages are the simplicity of use, the speed, and also the high throughput capability. It consists in exciting the fluorophore with polarized light. Depending on the fluorescence lifetime and the local rotation of the fluorophore, the emitted light will be different. So a fluorophore having a lot of motional freedom will emit a low polarized light, and the measured fluorescence anisotropy will be weak. On the contrary, a more constrained fluorophore will emit a strongly polarized light, and the fluorescence anisotropy will be high. So in practice, we have 96 well macroplates where we put our samples. Then we put the plate in a macroplate reader that will perform the analysis in about 90 seconds. To begin with, we used a 23-mer DNA aptamer targeting L-tyrosinemide, which is a model probe. The fluorescein reporter is attached to its 3' extremity, and we chose a MES buffer at pH 6.3 because of its very weak complexation features towards metal ions. And the slightly acidic conditions are particularly suitable for avoiding metal precipitation of metal hydroxides. So we performed metal ion titration experiments in presence and absence of targets by varying the metal ion concentration. For each metal tested, we compared the two obtained curves in order to eventually observe a signal difference between them. So we selected 40 micromolar of lead for the next experiments because that was this element that induced the most important shift of the curve. Next, we performed a dose-response experiment by varying the L-tyrosinemide concentration, so that's the red curve in this shimmy. And we observed a decreasing signal that we expected because the more we add targets, the more the metal ions fixed along the aptamer are displaced in favor of ligand binding. So the more the fluorophore is free and the more the anisotropy decreases. After that, we conducted some specificity assays by testing the aptamer with its non-cognate enantiomer, the D-tyrosinemide. Then by testing another aptamer with our target. And finally, by testing our method without metal ions in the medium. For each experiment, we saw that the signal remains constant, showing firstly that the method is specific, and also that metal ions are essential for the method to work properly. The next step, the next step consisted to improve our sensor by playing on two parameters. Firstly, we added magnesium at various concentrations because numerous papers have shown that magnesium has stabilizing effects on the active conformation of aptamer, promoting the target binding. So we thought it may improve the apparent binding affinity, but in return, we expected a reduction of the signal amplitude. So secondly, we reduced the MES concentration and consequently the sodium concentration because we assumed the ionic strengths may influence the maximal response obtained. So again, we made several dose response curves and we had a valuable compromise between apparent binding affinity, maximal response obtained, and also the dynamic range with 2.5 millimolar of magnesium. And we also determined a limit of detection of about 200 nanomolar. Then to assess the sensor quickness, we measured the fluorescence anisotropy during 10 minutes. And for each L-tyrosinemide concentration tested, the maximal response was obtained directly after the first read, so after 1 minute, demonstrating that the sensor possesses a high speed of responsivity, which is partly attributable to the very fast ligand exchange rate of the switching metal ions. After that, we wanted to apply our method for other small molecules and other types of aptamers, like a hairpin with aflatoxin B1 aptamer, a G-quadruplex with the ochratoxin A aptamer, and a three-way junction with the cocaine aptamer. So we made the same experiments as for the L-tyrosinemide model. This time we selected cadmium for making the dose response curves. And we saw that the fluorescence anisotropy decreases in presence of metal ions, whereas the signal remains constant when metal ions are absent, showing that our method works for each system. Of course, we made some specificity assay. I didn't put all the data on this slide, but all the specificity experiments were correct, and the sensor speed was about 5 minutes. Finally, we wanted to show that our method could work in real-life conditions. So we performed some dose response experiments with diluted urine samples spiked with either ethiocinamide or cocaine. And we found comparable results between the complex medium and the buffer medium, demonstrating that the sensor could detect such analytes in such a matrix. That's particularly encouraging as long as this biological fluid is quite challenging because it produces background fluorescence and also contains interfering components such as enzymes, phosphates, citrates, as well as variable amounts of metal ions. So we were particularly happy about these results. So to summarize, we were able to develop an easy to implement APTA sensor, which doesn't require expensive raw materials and with a fast reaction time. We also demonstrated the applicability to several small molecules and several aptamers with different secondary structures. The use of metal ions as transducers to induce a structural switching and thus a measurable signal is something that has never been done before. So we introduced something innovative. And since it's a method based on electrostatic interactions, we think that it could be adapted to other label-free platforms that report changes on the surface charge density of DNA, such as electrochemical or field effect transistor devices. So we published these results last year. And to note, this work has led to another work where we used some quencher transition metal ions to develop a novel fluorescence-based APTA sensor, and we published the results in February. So to finish my presentation, I would like to thank all the partners and all the people who contributed to this work in Nova team and also in hospital. And of course, I would like to thank you for listening. And if you have some questions, feel free to ask. Thank you.